So today I'm going to finish off the lecture series by talking about gene regulation in eukaryotes. So a lot of the groundwork for this lecture we've already been through. We've been through transcription control and what transcription is. We've been over the RNA polymerase II and the genes that code for messenger RNAs, and we've looked at some of the sequences in the promoter that are important for the assembly of that basal transcription machinery we talked about. But we'll sort of, and last week we talked about gene expression in prokaryotes. So last week we were looking at gene regulation in bacteria, and we were talking about the operons, the regulation of multiple sort of proteins through an operon, which has a variety of gene sequences hooked up to a single promoter. So what we're looking at today is pretty much building up on what we talked about last week, rather than here's a whole new set of things. It's like this is building up on what we have already discussed. So hopefully that makes things a bit simpler. Okay, so what are some of the differences that we need to consider when looking at gene expression in eukaryotes? I've kind of raised a couple of these at you before. I think I raised issues before about how there's just so much more DNA in a eukaryotic cell than you would anticipate based on a prokaryotic cell. Okay, so I think also the number of genes is fairly, there's a lot more as well. We've got maybe 30,000 genes. So I think if you take a prokaryote and look at how much DNA it has for the number of genes, I think a prokaryotic genome is basically made up of gene sequences. There's not a lot of other stuff in a prokaryotic genome. Okay, there aren't introns. I don't think there's that many, intervening sequences. Whereas when you get into eukaryotes, not only are the genes much bigger because they've got introns, and introns often make up more of the gene than the actual exons do, but out of the genome, only about 4 or 5% of the genome is actually genes, actual sequences that you would consider to be a gene, whether it's intron or exon. Okay, so we've got a lot more DNA in eukaryotes that is doing other things. Okay, the byline when I was an undergraduate student was that most of our DNA is junk, and buried in there was some genes, But we now have a much better understanding of the other rest of the genome. But what I'm trying to say to you is that when you go from a prokaryotic system to a eukaryotic system, it's not a linear increase in the amount of DNA. It's a huge increase in the volume, the amount of DNA that the cell has to deal with. So in eukaryotic cells, we have packaging of DNA into a different form, into chromatin. Okay, so we've got the sheer volume of DNA to deal with, and also we've got more genes. So we still need to be specific for which genes are being regulated at what point in time. Okay, so in bacteria, the RNA polymerase has access to all of the promoters, because in prokaryotes the DNA isn't packaged up in chromatin. Okay, in prokaryotes the DNA is fairly accessible. Okay, it's a smaller amount of DNA. It's not tightly packaged. It's fairly accessible. So effectively, then the RNA polymerase can bind and initiate transcription at some low level of efficiency, and then in the addition to that, I discussed to you some activators and repressors to make the polymerase more efficient or to block transcription. Okay. When we get to talk about today's lecture, when we start to talk about eukaryotic genes, the promoters are generally stronger, and you can activate gene expression to high levels in eukaryotes. But also the ground state, if you like, the general level of expression of genes in eukaryotes is they're transcriptionally inactive. Okay, so the ground state, in the absence of repressors and activators, then the ground state is that eukaryotes, they're said to be restrictive. Okay, so genes are off in eukaryotes because of DNA packaging. Okay? Whereas in prokaryotes, we don't have DNA packaging, and the DNA is accessible for the RNA polymerase to bind to it. Okay? So that's a big difference. So in eukaryotes, even though we have repressors, we're typically talking about gene activation, turning genes on, because of the packaging of DNA, genes are generally off. Right? Because don't forget, in prokaryotes, we don't have a cell nucleus, and we don't have packaging of DNA with proteins into higher order structures. Okay, so some distinguishing features of gene regulation. So the first one, as I've just been talking about, is that the access to the promoter is restricted by chromatin. Okay, so access to, what does that mean? Access by what? Transcription activators, the things, and the polymerase and all of the basal transcription machinery. It doesn't have particularly good access to the promoter regions of the genes, okay, unless the chromatin is accessible. Okay, so transcription activation is associated with changes in chromatin. That statement seems fairly innocuous, and it is. But when people first were studying gene transcription, they were thinking about turning genes on and off and the polymerase and everything. And then another bunch of scientists started to study packaging of DNA, and they started to identify histones and nucleosomes and all of that sort of thing. But they didn't think of DNA packaging as a level of control in gene expression. They thought of it as different things. But now it's become apparent that packaging and regulation are the same thing. Okay? Again, it's very difficult to separate the two, because when DNA is tightly packaged, it can't be expressed. And when DNA is loosely packaged, it can be expressed. Okay? So access to the promoters is restricted by the chromatin, and transcription activators are associated with change in chromatin. Okay? And that's related to gene expression. Okay? And this is not the case for prokaryotes, because in prokaryotes, DNA isn't packaged as chromatin. Right, and I'll explain what chromatin is in a few slides' time. Again, last week I was talking about proteins that regulate DNA expression, gene expression, and I talked about the lac operon, and we had the green-coloured cartoon protein that was the activator, and the red protein that was the repressor, following the traffic light kind of cartoon. So in eukaryotes, we also have positive and negative regulatory mechanisms. In other words, repressors and activators. But predominantly, positive mechanisms predominate. Okay, so genes are turned on because generally in chromatin it's hard to access them anyway, so you have to switch them on. So we talk mostly about gene activation rather than about turning genes off, because the ground state is off, so you have to turn them on. Eukaryotic cells have a larger, more complex protein regulatory network. So effectively, because we've got more genes to regulate, we have more proteins involved in gene regulation. Okay, we've got more transcription factors. Okay, we've also got other co-regulators and other proteins involved in the process as well. Okay, so and a point I've already mentioned, there's no need to really go into this again, is the fact that in eukaryotes we have a separation of transcription and translation, because we have a nucleus, so transcription occurs in the nucleus and translation occurs in the cytoplasm, whereas in prokaryotes transcription and translation are coupled and can happen simultaneously. So we have this separation in space and time in eukaryotes. But I guess what this means is that there are many more opportunities to regulate the process of gene expression in eukaryotes because of this separation. So if something is happening at a later time in a later place, then there are different regulatory mechanisms you can put in place to regulate the expression. But we're not going to talk too much about those today. So what we are going to talk about today is this idea that chromatin structure plays an important part in gene regulation. Okay? So when we're talking about chromatin structure, so chromatin refers to a complex of proteins and DNA. That's what chromatin is. And always, your DNA in cells is associated with proteins. So always, your DNA exists as chromatin rather than as naked DNA or purified DNA. It's a complex of proteins and DNA that come together. Okay, so that's how DNA exists in our bodies. When we talk about packaging of DNA, we talk about different levels of packaging. We've got very simple packaging, and then that folds up to a second level of packaging, and then that refolds up into a third level, and it keeps refolding until you get to a chromosome, which is the highest order of DNA packaging that you can observe during mitotic, during mitosis. So at the level of the gene, we have packaging of DNA, and at the level of the chromosome, we have the whole thing. You can see the whole thing packaged. So there's various levels of packaging between nucleosomes, which are the first order of packaging, up through to chromosomes. So nucleosomes are said to be the building blocks of chromatin, and nucleosomes are a complex of histone proteins and DNA. So I don't know if you've come across histones before. So a nucleosome is made up of histones. Now there are 8 histones in a nucleosome. The histones are called H something. H indicates histone. We have H1 is not involved in the nucleosome. H1 is a loose protein that binds in the vicinity of the nucleosomes. Okay, so put H1 out of the picture for now. What a nucleosome is made of, it's made of 2 copies of histone 2A, 2 copies of 2B, 2 copies of histone 3, and 2 copies of histone 4. So we've got 4 proteins, each one occurs twice, and those 8 proteins make up an octamer, and that makes up a disc-shaped structure. So it's like a disc, and I've got some diagrams that show what a nucleosome looks like. So the proteins of a nucleosome are histone proteins, and there are 8 of them in a structure which is a bit shaped like a disc. Okay? It says here small basic proteins. Basic doesn't mean simple as opposed to complex. Basic means charged. Okay? So they contain more basic amino acid residues than you would expect. Okay? So they're rich in lysines and arginines. Okay? So if you remember proteins and genes, lysines and arginines are the amino acid side chains that have positive charges on them. Okay? They've got NH2, NH3 groups on them, and they're positively charged on their side chains. So basic proteins means proteins rich in lysine and arginine residues, and histone proteins are rich in those. And they come together to make up these nucleosomes, and there are 8 nucleosomes, 8 histones in a nucleosome. So we've got DNA tightly wrapped around a nucleosome, or should I say tightly bound to the nucleosomes, okay, because of the charge difference. So the RNA polymerase, which also wants to bind to DNA, it can't bind to DNA easily when the DNA is bound to histones. So it's a simple fact that the DNA is wrapped around and inaccessible because it's bound to these histone proteins, and then higher orders of structures makes it even more obscure and more difficult to get to. So the transcription machinery needs to deal with chromatin structure before it can access and turn on a gene for gene expression. So as a result of chromatin structure, eukaryotic genes are generally expressed at low levels in the absence of proteins that can unravel the chromatin and allow transcription to occur. Right? So, like I said, chromatin is a complex of proteins and DNA. The the protein that occurs in the highest amount, if you like, are the histone proteins. But there are other proteins that constitute chromatin. One of the ones we'll see in a few slides' time in the pictures is HMG proteins, high mobility group proteins. We'll see those, and there's probably there's others as well. But histones are the most common, and histones are also highly conserved. So if you go back to the simplest eukaryotes, typically we'd say yeast is a simple eukaryote. So yeast has a nucleus. There's a few introns in some yeast genes. And it's got similar fundamental processes of cell cycle regulation and DNA metabolism that we have in higher eukaryotes. And when you look at the histones of these simple unicellular eukaryotes, histone proteins are highly conserved. They're some of the most highly conserved proteins there are across eukaryotes. And as soon as you see something that's highly conserved, what that means is that, well, if you make changes to histone proteins, it affects the cell's ability to survive. So any changes and mutations that occur in histone genes, those cells don't survive. So histone proteins are very important, and any changes to the histone genes is detrimental. So they've been highly conserved over evolution, which tells you straight away that that histones and nucleosomes play very important roles in a cell. Okay, so histones are highly conserved. They're basic proteins, and they assemble into these octameric complexes, this disc-shape structure that I'll show you. And each histone contains each of the 4 histone subunits. Now, when you've got your disc, if you like, your disc of histones, DNA wraps around the outside of the disc. Okay, and it's about 2 wraps. It goes about once and maybe a bit more. So it almost wraps around twice. So if you count the base pairs, it ends up being 146 base pairs of DNA, gives you the wrapping around the histone proteins. And then between the next one nucleosome, and the next nucleosome, there's a linker DNA. So there's a region of DNA. It could be 100 base pairs, it could be 200 base pairs. It's not as fixed, and then the DNA sort of we just have that repeating unit of nucleosome, bit of linker DNA, nucleosome, bit of DNA, nucleosome. We just have a string of nucleosomes across the genome. So nucleosomes are the first stage in packaging of DNA into higher DNA packaging, and the highest level of DNA packaging will be what you see in a chromosome during mitosis. So this is an electron micrograph showing DNA from a eukaryotic cell. Now, if you think back to, it was probably the last lecture, I showed you an electron micrograph that showed DNA, and you could see the messenger RNA being made from the DNA, and then you could see ribosomes attached to the messenger RNA. Do you remember that micrograph, anyone? No, it was one of these little dark blobby things with, No? Okay. Well, contrast that picture, if you can find it, to this one. Okay? This is showing DNA in eukaryotic cells. Okay? What you didn't see in that other micrograph was any DNA was just a string. There was no blobs on the DNA string. It was just a string. Okay? Because in bacteria you don't have nucleosomes. Okay? What you see here is packaging of DNA. So this is a string of DNA here, and here are the nucleosomes. And it's not clear from the structure what is actually happening there, but people have worked out that these are where the octamer of histone proteins are. And if you look at the top diagram here, this is the cartoon that is drawn to represent these beads on a string. Okay? So I think these beads on a string, they probably date back 30 years now. I'm not sure when they were first, people first started to identify and think about nucleosomes in a meaningful way. But for at least 30 years, people had known about these beads on a string. It's actually, and what this cartoon is showing you here is that within this disc, this is that disc shape, these histones here, you can see the DNA wrapping around about twice, and then a linker region, and then wrapping around about twice, then the linker, and so on and so forth. So it took, actually, it was more than 30 years ago they discovered this, because it took 30 years for them to progress from that level understanding to this level understanding. Okay? So on the 30th, I think the 30th anniversary of the discovery of nucleosomes, they finally got together the X-ray crystallography structure of a nucleosome. And I've included this, which isn't in the textbook, because it was one of those groundbreaking moments in science when you just saw that paper and just went, wow, that is really fantastic. This is great science, and it just makes stuff really clear. Because when you're trying to understand processes, it's very hard to understand processes when you're looking at these kind of structures here. This is telling you a lot, but it's proposing more questions than answers. It just gets you thinking about what are these things here and what's going on. Okay, so this is the X-ray crystallography structure of a nucleosome, and in the center here you'll see your 8 proteins. So there's probably 4 colors here indicating 4 proteins, and each one occurs twice. So there's your 8 proteins here. And then if you do if you look at this, I've got a side view as well. This is taking that structure and flipping it on its side. This is the side view of that disk. Okay, so if you look at the DNA, you can see the DNA wraps twice around the disk, and like I said before, it's about, you don't need to know it, but it's about 146 base pairs of DNA. So between the inside of the DNA here and these proteins are strong contacts which make the nucleosome very stable. Right? What became apparent from this this structure, which nobody anticipated, and everyone just went, wow, and kind of bowled people over, it doesn't look like much, but if you look closely, you can see these little strings here. See these little strings sticking out from the nucleosome? These are the I think they're the N-terminal domains of the proteins. So every histone in that disk has its N-terminal domain protruding out from that nucleosome. Okay? And it's this tail here that's rich in those lysine and arginine residues that we were talking about. And these tails here suddenly give us a clue on how the nucleosomes are packaged, and what controls nucleosome packaging is these bits of string that are sticking out that can be used to package and unpackage the DNA around these nucleosomes. Okay, so this is the structure of a nucleosome at sort of high resolution level. This is the textbook version of the nucleosome. Obviously, it's taken from that paper. They probably use the same color coding and the same structure for the protein here, and they've emphasized the histone tails in this cartoon here, which is important. They've labeled the histones, so there's 2 copies of the histone H2A, 2 copies of 2B, 2 copies of 3, and 2 copies of 4. And for each of those proteins, you have these histone tails protruding out of the nucleosome. So the histone tails don't contribute much to the strength of binding. The strength of binding is on the inside here of the disk, or of the DNA, is where the strength of binding between the proteins and the DNA is. But the histone tails, they form intramolecular contacts with adjacent nucleosomes. And it's these tails that can be modified to determine how strongly it's going to contact the next nucleosome. So if they're in strong contact with each other, they're strongly interacting, then it's very difficult for a DNA polymerase to come along and get in there and bind to that DNA and to then start to activate gene expression. Okay, it's a difficult thing to do. But if you can somehow manipulate these nucleosomes so that they're more open and more accessible, then the polymerase and other factors have got a chance of getting in there to turn on gene activation. So this is the repressive state that DNA is in a eukaryotic cell. It's generally switched off because it's packaged in chromatin. And the most common form of chromatin are these are histone proteins, and this is a nucleosome made up of histone proteins and DNA. Again, this is the cartoon from the textbook, just showing these N-terminal domains of the histones. I don't know why they've drawn 3 here, but anyway, it just shows how they can somehow interact to modulate the packaging of these discs. So, okay, so we're talking about packaging of DNA, and I've said the highest order of packaging of DNA is the chromosome, shown here. So if you look at the scale, this is looking at about a 1,500 nanometers. So, so these are quite large structures, obviously huge structures on a molecular scale. But working backwards, we're going to be looking at gene expression that occurs at this level here, predominantly at the level of the nucleosome. Okay, even though high level packaging has an effect on expression, the more you move from this naked DNA through these higher order packaging, the less is known. So we know an awful lot about the structure of DNA. We now know quite a lot about nucleosomes because of the X-ray crystallography structure of nucleosomes. We're starting now to understand this 30 nanometer fiber. And then we've got electron micrographs of these extended loops and things, but we understand it less because it's much more difficult to study at a molecular level. And you get big things. Okay. So this is the double-stranded DNA, 2 anti-parallel strands. It's about 2 nanometers long, and here you've got, we've got 10, 20, 30, 40, 50 base pairs of DNA or so. Now, this sequence of DNA here is represented in the nucleosome sort of diagram by that region there. So that's a little bit of the linker region, all right, shown at the level that you're probably used to looking at DNA in this sort of cartoon. So at about this level here is just a run of a couple hundred base pairs of DNA. Okay, and then you've got your 146 base pairs wrapped around your nucleosome. And in this diagram here, it's showing 6 nucleosomes. So if that's about 150 and that's about 100, then that's 250 base pairs. So that's 200, 50, 500, 1,000. That's about 1,500 base pairs of DNA in this little structure here. Okay? And then in the next diagram, we're looking at this 30 nanometer fiber or 30 nanometer filament. And you can see these 6 nucleosomes here, shown here, organized as part of this helical structure. So again, that helical structure there has about 1,500 base pairs of DNA, and you've got 1, 2, 3, 4, 5 lots of those, so I don't know, 7,500 base pairs of DNA wrapped up in that little section of fiber there. And then you go to the next stage, where all of this section here is now represented by this little bit here. So this is another cartoon. But again, it's based on lots of electron micrograph structures where you can see these big fibers of DNA looping out from some sort of support material shown down here. Okay? And later on, when we're talking about gene expression, and we've already talked about this in previous lectures, we talk about the concept of you've got a promoter for a gene here, but tens of thousands of base pairs away from the promoter, either upstream or downstream, there are other regions of DNA that affect the expression of the gene. Okay? So maybe one of the ways these distant regulatory regions can have an effect on the promoter is through this level of chromatin packaging that we see in these diagrams here. So for instance here, you've got this little looping structure, and there are regions of DNA that are separated by tens of thousands of base pairs that are in very close proximity between those two structures. Okay? So maybe this is one way that distant regulatory elements can come into close proximity to promoter regions of genes. Okay? Otherwise, I don't know how you visualize how a region of DNA 50,000 base pairs away can have an effect on a promoter. It's almost a nonsensical concept. There has to be a mechanism, a way of it coming into close proximity. So maybe this is one of the ways, and I guess you'd call this kind of like a looping of the DNA to bring these distant regions together. from there we go on to this more condensed structure, which again is understood at various degrees, but not at high resolution like we understand nucleosomes and normal DNA. And then, so this region here is a small section here, and then again we've got the packaging into the chromosome. So when we talk about packaging of DNA in eukaryotes, we've got a lot of DNA associated with these histones and other proteins packaged in various degrees. So it's typically it's highly packaged in cells. Okay, so the RNA polymerase II and the polymerases and all that basal transcription machinery has to get in through this packaged DNA to access the promoter to turn the genes on. Okay, so in this sense, the genes in the genome are in a restrictive state and they're not easily expressed. So we have positive mechanisms that turn on gene expression. That's the sort of narrative the textbook gives you. So I guess the thing to look at now is what are some of the factors that affect packaging, because when you loosen the packaging, you allow gene expression to occur. So nucleosomes are said to be remodeled. Okay, now there's different ways you can remodel nucleosomes. The textbook covers a couple of them, but one of the ways of remodeling is to physically shunt the nucleosomes away from the promoter. So it's like a goods train stuck on the tracks. You can just shunt it away and move it away from the station so you can access the station. That sort of thing. So there are mechanisms in the cell that have been uncovered that include the hydrolysis of ATP providing energy to shunt these nucleosomes and sort of wind them away out of the promoter regions of genes. So one way is to do that, and then what we discover is that active promoters, ones that are actively expressing genes, these active promoters are typically open regions of DNA, and so the nucleosomes have somehow been shifted away from the promoter region. Okay, and then this allows the access of transcription factors into the promoter region. Another way of accessing the DNA in the promoter region is to make the association between the histones and the DNA, to make it looser. Okay, so tightly bound DNA around a nucleosome is much harder to access than loosely bound DNA around a nucleosome. Okay, so not only can you shunt them away and completely remove them, but you can also make them less compacted, more open. Okay, so the nucleosome might still be there, but it's a more loosely bound nucleosome which can allow other proteins to bind. So we also have post-translational modifications to histones. Now, I've used the phrase a couple of times in these lectures, post-translational modifications of proteins, typically to turn proteins on or off through phosphorylation, or to target proteins, I haven't talked about this, but to target proteins for degradation through ubiquitination, or proteins can be methylated. So different side chains of the amino acids can be modified after the protein's been made. Okay? And one of the important modifications that occurs on histone proteins is a process called acetylation. So an acetyl group is a simple 2-carbon structure which is added on to some residues in the protein. The residues that are modified by acetylation typically are lysine and arginine. So it's these basic side chains that we've talked about. We know histones are rich in lysines and arginines, and part of controlling chromatin structure is by modifying these lysines and arginines that are in the histones. So one of the enzyme activities that's involved in this process is something amusingly called a HAT, okay, a histone acetyl transferase. So these HAT activities, okay? And when you modify these lysine residues, you cause a decondensing of chromatin. So in other words, a loosening of chromatin. So a simple way to think about it, and then sort of an easy way to think about it, is if you think of the sugar phosphate backbone of DNA, the phosphate under physiological conditions is negatively charged. You've got those O- groups on the phosphates. Okay, so you've got a strongly negatively charged DNA backbone, and then you've got these histones with these big tails that stick out of the nucleosomes, which are positively charged with all those lysine and arginine side chains. So all of those positively charged residues can bind to the negatively charged DNA. And then when that happens, people think, well, it makes that nucleosome much more tightly packed and packaged, and it's much more difficult to displace so that the RNA polymerase can get in there. Okay? So one way to turn on a gene is to bring in these histone acetyltransferases to add acetyl groups to those positive charges on lysines and remove those positive charges from lysines in that process. And therefore, these histone tails bind less strongly to DNA, which make it a loosely packaged nucleosome. Okay? So this results in decondensation of chromatin, and it provides access of DNA binding factors, these transcription factors, to the DNA. Okay? There are some proteins contain these domains called bromodomains, and they can bind to these acetylated histones, further opening up the structure. So this is a fairly instructive cartoon. It's quite a useful cartoon. The first thing you notice from this cartoon is that in this histone tail here, you've got the residues have been numbered. So you've got residues 1 to 20 here. So we know there are 20 amino acids. So there's a 1 in 20 chance of there being a lysine residue, okay, just by chance alone. But there's 4 of them here, okay? And again, you've got 4 of them here. So lysines are highly overrepresented. It's about 4 times as many lysines as you'd expect if there weren't other things controlling the amino acid sequence. And these lysines can be acetylated. So these red blobs here are lysine residues which, when you pull them out of cells and you sort of do your chemical analysis, well, it goes, well, these lysines have been acetylated. Okay? And if you can then look at genes when they're on and off, you can look at whether lysines are acetylated or not acetylated. And clearly make observations that acetylated lysines is associated with gene expression. Okay? There's other modifications listed in this cartoon here as well. We've got your typical serine residue that's been phosphorylated, and then you've got other lysines and arginines that have been methylated or some other modification. But predominantly, the one that we'll talk about because it's the most common is the acetylation of lysine residues on these histone tails, shown by these red dots here. Okay, and the activity that carries out acetylation is called a histone acetyltransferase. Okay, so these HAT activities are histone acetyltransferases, and once you've acetylated those lysine residues, you're allowing transcription to go ahead. And it'll probably come as no surprise to you that in a couple of slides' time, I'll talk about another enzyme activity that removes acetyl groups from lysine residues. Okay, so whenever you've got an on switch, you have to have an off switch. So if the on switch is acetylation of lysines, then the off switch is the deacetylation of lysines. So histone modifications can also cause chromatin to become tightly closed, and that's the activity I'll be talking to you about, which is the removal of acetylated acetyl groups through what we will refer to as a histone deacetylase. And other examples are methylated histones can be bound by proteins that also help to condense chromatin. So here's a little, again, a little cartoon from the book, and here's our HAT protein activity here, our histone acetyltransferase, which causes acetylation of these tails, and these red blobs here indicate that there's been some covalent modification, there's some acetylation. Okay? And once you acetylate these side chains of the histones, you can see that the chromatin here is drawn in a much more open and loose format than it was here. So chromatin can be described as two types of chromatin. You can have heterochromatin, shown on the left side, which is tightly compacted and not transcribable. You can't transcribe heterochromatin. And on the right side here, you've got euchromatin. Okay? And there's various states of euchromatin, and you can see here euchromatin is the open form of chromatin with these chemically modified side chains of the histone proteins. One of the important activities which is important in gene expression, in turning genes off, is something called a histone deacetylase, okay, which is shown here. And this is the opposite of a histone acetyltransferase. It simply takes off those modified acetyl groups, and it returns those lysines to their positively charged form, and it allows the histones and nucleosomes to tightly package up. Okay, so chromatin can be in two forms. One form is very difficult to transcribe from, or express genes from, and the other form is open, and then we can start to get transcription activators in there to help turn genes on. Okay, so in this form, you can see it's very difficult for the polymerase to get access to the promoter region of the genes. I can see some of you are so excited that you're texting to your friends. I'm at this really great lecture, and he's talking all about transcription, and it's really fascinating. But I know what it's like, don't worry, I'm not trying to hassle you. Okay, so we're moving on now to... So I've been saying to you that tightly packaged chromatin is called euchromatin, that the most tightly packaged chromatin you find in the genome, about 10% of the genome is really tightly packaged, but it's not regions of the genome where you expect to find gene expression. So there's centromeres and telomeres, so the center of the chromosomes and the ends of the chromosomes where there aren't genes. It's more to do with pulling chromosomes between cells and maintaining the ends of chromosomes. They're tightly packaged with these in euchromatin. And in other regions of the genome, the other 90%, you've got various states of packaging, and it's said in that diagram it's packaged as euchromatin. So there's a nice little cartoon here again from the textbook which shows now gene expression in the presence of euchromatin. Okay, so in this example here, you can see that the acetyl groups have been added onto these histone proteins. So effectively, because it's been acetylated by histone acetyltransferases, that's already happened. This chromatin is open for transcription. Okay, and whilst it's open for transcription, there's been the binding of the RNA polymerase has been able to get access to the gene. Again, this bendy arrow here indicates the start of transcription, and the on indicating that the gene is on, so it's being transcribed. We've got bendy elbow arrow here and the on. And what you can see here is the binding of transcription factors, which are activators, which are bound directly to the DNA. Okay? And here, if you recall last week when we looked at prokaryotes and gene activation in prokaryotes, we had a simple traffic light system. We had the green activators and the red repressors when we looked at the Lacopron. And we went through to describe what those proteins were in detail. Again, the textbook's using the same drawing here, and we've got a green protein here which is an activator. Okay? And again, the activator is facilitating binding of the RNA polymerase to activate transcription. Okay? And again, in terms of nomenclature, we've got something called a co-activator. So co-activators don't bind to DNA. They bind to the proteins that are bound to DNA. So here's the transcription factors bound to DNA, these activators, and then in between these two transcription factors, you've got a co-activator, which is protein-protein interactions. The co-activator isn't bound to DNA. Right? And then, so this is the open state where a gene can be expressed because the histone acetyltransferases have acetylated those lysines and things on the histone tails. The next step in shutting off a gene and turning off a gene would be some repressors get involved to bind. Now, I guess the way of rationalizing what's going on here, these repressor proteins have a stronger affinity to bind to DNA than these activator proteins. So that these repressor proteins end up displacing the activators because they've got a stronger affinity for DNA. Okay? So these strongly binding repressor proteins have displaced these transcription factors and co-activators, and they've tethered in these other proteins, which we just call co-repressors because they're doing different things. And this co-repressor now is not facilitating, the polymerase to bind and to transcribe. And then what the co-repressor can do, it can bring in another activity, would you believe, a histone deacetylase. Okay? So these transcriptional repressors have now tethered a histone deacetylase into this gene. Okay? And that's going to remove these acetyl groups because it's a histone deacetylase. Okay? It's the opposite activity of a histone acetyltransferase. So the deacetylase will take away from these lysine side chains, it will take away those acetyl groups. Those lysines become positively charged again, like a normal lysine, and then they're able to more strongly interact with the DNA, causing repression of that gene and turning off of that gene. And then you end up with this tightly packaged gene and promoter region tightly packaged in euchromatin that's difficult to access. So it's quite a nice simple story now, going through those slides, of looking how genes are actually turned on and turned off in eukaryotes and how it's linked up to packaging. And this narrative makes a lot of sense to me as a mechanism of turning genes on and turning genes off. And it's kind of has come out of many, many years of research into protein DNA interactions, everything from those beads on a string through the X-ray crystallography structure of the nucleosome, and then identifying a whole bunch of other things. Okay, so I've been talking about this concept, and I've got some slides that talk about this as well coming up, that the regulation of eukaryotic genes is affected by regions that are very distant from the promoter. So in the slides I'll show you, it can be up to 50,000 base pairs away. You can get regions of DNA that are thought to be involved. Okay? Now, some of the evidence that supports this notion comes from, it's being talked about here. So this slide is talking about hypersensitive sites that correspond to binding sites for known regulatory proteins. So what does that mean? So in the previous lectures, I've been talking to you about nucleases. We've got endonucleases and exonucleases, and we talk about those. Okay? Exonucleases digest in the ends of the DNA strand. Endonucleases can digest in the middle of the strand. Okay? So people have done studies with endonucleases. Okay? Now, because the chromatin is tightly packaged, the endonucleases can't access DNA unless it's open. And when the DNA has been opened up, the endonucleases can access the DNA and digest the DNA. Okay? So it's very easy to do experiments, and you look for regions of DNA that can be easily digested by endonucleases. Okay? So if you can get a cell state or cell type that's expressing a gene in a really strong way, you look at the hypersensitive sites where the endonuclease can digest the DNA. You then do the experiment where that gene is switched off, and again you look for hypersensitive sites away from the promoter. And you can find regions of DNA that correspond to being hypersensitive when the gene that's 50,000 base pairs away is active, and that are not hypersensitive when the gene is off. So there's some very clear evidence that says DNA at distant regions is involved in regulating particular genes. Okay, so that's one of the many types of evidence that support this notion of upstream or downstream regions that are very distant can be involved in gene expression. Okay? And maybe trying to rationalize that data, you can either throw your hands up and say, that's absurd. How can a region of DNA 50,000 base pairs away have any effect whatsoever? You're talking nonsense. Or you can say, yes, it's because we have looping of the DNA, and through that looping and that high order structure, maybe those regions of DNA can somehow come into close proximity. All right? So there was quite a debate in the science community 10, 15 years ago about people who trying to rationalize how these hypersensitive sites are involved with gene expression, how the DNA can come together, and it was, you know, there's often areas of science that are hotly debated. And it's, if you're doing science and you're involved in an area, it's often really good to contribute to the arguments and to have these open discussions about what's happening. So some people believe that looping was a thing, and that DNA could loop and come into contact based on, you know, high order structure, and some people have other theories of why or how these hypersensitive sites can be transcriptionally related. But when the textbook is clearly describing looping as a thing, so we'll stick with that narrative. So these hypersensitive sites that are very distant from the promoter regions, they become open and hypersensitive when the gene's on, and they become not hypersensitive when the gene is off. And at these hypersensitive sites, if you look at the DNA sequences, it also contains binding sites for some of the same regulatory proteins that are binding at the promoter region of the gene. Okay? So, and it's thought that the open structure of these hypersensitive sites, the absence of nucleosomes, allows these transcription factors to bind at the hypersensitive sites. I've also talked to you about the ribosomal RNA genes. Remember the ones that are transcribed by RNA pol 1? What I was saying to you in that other lecture is that the ribosomal RNA genes are highly expressed pretty much all the time because we always need ribosomes in the cell. So these are genes that are highly expressed a lot of the time. And when you look at how open the regions are around these ribosomal genes, they're pretty much they're loosely packaged, meaning that it's not highly condensed promoter regions, and the transcription factors have got access. So nucleosomes are almost entirely absent at these highly active genes for ribosomal RNAs. Again, further evidence that packaging of DNA is intimately associated with gene expression, or the lack of packaging is associated with gene expression. Also, people have decided, have realized that transcriptionally active chromatin tends to be deficient in histone H1. So histone H1 has been absent in those cartoons I've been showing you of the nucleosomes, because we've got the 2A, 2B, 3 and 4 are the histones that are involved in the actual disc that is the nucleosome, but the linker region between the nucleosomes is where histone H1 binds. So histone H1 is thought to be involved in condensing the nucleosomes, and when histone H1 is absent, the nucleosomes are thought to be less tightly packaged. So transcriptionally active chromatin tends to be deficient in histone H1, which binds in that linker region between the histones and the nucleosomes. Okay, so we've looked at the fact that acetylation and deacetylation of histones affects the degree of packaging, how tightly packaged they are. So acetylation and deacetylation of histones figures prominently. So to activate chromatin for transcription, it becomes acetylated. Okay, and to inactivate it, to turn them off, it becomes deacetylated. Okay, so just think of it as the positive charges on the lysines tightly bind to the DNA, and that's off. And then when you acetylate those lysines, it's not positively charged, doesn't interact strongly with the DNA, so it can be more loosely packaged. Okay. So when transcription is no longer required, then these histone deacetylases are brought into the story, and they tend to allow chromatin to become more tightly packaged, which is what we were looking at in this diagram here, when we had these repressor proteins bringing in the histone deacetylase to turn off gene expression. Okay, so I'm just recapping on some of the points on these diagrams here. So in the eukaryotes, I was saying that we have positive gene expression. So whenever you want a gene, it's not a matter of turning it off because it's off. It's a matter of turning it on. Okay. So we have positive regulation of gene expression at eukaryotic promoters. There are 3 different polymerases in eukaryotes. They have little intrinsic affinity for promoters. Okay, so transcription to be strong and for there to be a strong association, you need these activated proteins to help the polymerase bind strongly to the promoters. And so we have a bunch of transcription factors that are important. Okay, now again this gets back to this idea of we've got a lot more DNA, we've got a lot more genes in eukaryotes, but we don't have necessarily that many more transcription factors to account for the excessive DNA and the larger number of genes. We still have more transcription factors, but what we see in eukaryotes is effectively what we call combinatorial control of gene expression. So we get combinations of transcription factors working in tandem that lead to specificity of gene expression. It's quite a convoluted sentence. So when we looked at the trip operon, we were just showing one activator for one operon and one repressor for that one operon. So it's quite a simple case of here's a gene, here's the repressor of that gene, and here's the activator of that gene. With eukaryotes, where we say, well, here's the promoter, and here's a bunch of transcription factors that are involved in its regulation. And it's the combination of transcription factors that turn a gene on or off. Okay? So just refreshing on the RNA polymerase II, which is the polymerase that is making, that's transcribing these messenger RNAs. Typically, these promoters have a Tata box initiator sequences which are recognized by these parts of the basal transcription machinery. And in addition to these proteins binding at the promoter, often these promoter regions have these enhancer sequences. So enhancer sequences equate to these hypersensitive sites that are found in other regions of the gene, of the chromosome, or regions that are just distant from the promoter. So an enhancer is a sequence of DNA that's away from the promoter that can enhance expression from that promoter. Okay? So we've talked about promoter regions in genes. We also now are introducing this concept of enhancer regions, and enhancer regions are often these hypersensitive sites which are upstream and quite distant from the promoter. When we're looking at yeast, which is a simple eukaryote where a lot of transcription regulation was first understood, we don't maybe have enhancers, but we have upstream activated sequences. So enhancers can be many thousands of base pairs away. In yeast, these upstream sequences are much closer to the promoter, hundreds of base pairs away. Another reason of rationalizing why we have enhancer regions in eukaryotes, given that we're discussing multiple transcription factors that are required, so it's combinatorial in that regulation, at the promoter region itself, there simply isn't enough physical space to bind all of the transcription machinery and then to bind all of these other transcription activators and things. So basically other regions of DNA have to be included so that other transcription factors can bind, and then it's the combined effect of these different transcription factors that can turn genes on and off in specific ways. So not all of the binding sites that control gene expression tend to be adjacent to the promoter. Some of them can be away from the promoter at these enhancers or these upstream elements. So transcription factors must be able to have their effect at a distance. So things that are bound thousands of base pairs away must still be able to affect gene expression based on the data, the experimental data. So here's a little cartoon that summarizes what we've been talking about. So here's the promoter region of a gene, here's the Tata box binding protein, and some of these general transcription factors. So these transcription factors here are necessary for every gene. Every gene that's been turned on will have this group of proteins bound at its promoter. Okay? So they're called the basal transcription machinery or the general transcription factors. And you can see here the open DNA, and an RNA is about to be made. We've got breaks in the DNA sequence here, indicating that it's not a linear scale. So you've got a linear run of nucleotides here with an enhancer bound, but then some distance away, whether it's hundreds of base pairs or thousands of base pairs away, you've got other sequences which are known to bind transcription activators that are important to regulate what's happening here. And the same way, if you go downstream, either in the gene itself or thousands of base pairs away from the gene in the downstream direction, you can see regulatory sequences that bind transcription factors that affect expression of that gene. Okay, so this is one of those areas that's poorly understood, but the evidence is clearly there that these distant regions have an effect. And the simplest way to think about it is the looping model, where those regions of DNA are somehow looped around and in close proximity to the promoter, so that you can get interaction between those proteins. So these are enhancer sequences. Like I said, they can be hundreds of thousands of base pairs away from the transcription start site, or as that diagram showed, even in the gene itself. And then here we have the title of this one here refers to the yeast example on the bottom. So here's the start of transcription of the +1 with our bendy arrow. And then if you go 100 base pairs or even 250 base pairs away, there are sequences of DNA here that affect what's happening at the promoter. So if you're in a yeast genetics lab and someone says, go away and study regulation of this gene, which is the situation I was in when I was working in a yeast genetics lab, when you clone and chop up your gene and your thing to put it into a plasmid, you make sure you include at least 1,000 base pairs of the promoter, because otherwise you're going to miss important elements that are involved in the regulation of expression of that particular gene. Okay? If you're a mammalian molecular biologist as opposed to a yeast geneticist, whatever, and if you're working with human cells, then when you take your promoter and your gene and put it into a plasmid to study it, you're not going to capture the expression of that gene unless you include these distant regulatory elements. Okay? So it's a much more complex process to study the expression of a gene in eukaryotes, because you've got to include these distant regions here. So if you think, I don't know, you probably don't know, if you go through the history of understanding gene expression in eukaryotes, there's a cluster of genes that was probably the best understood genes, and they were the globin genes. Okay, so we have, the reason globin genes were chosen is because, this is not part of the lecture, you don't have to write this down if you don't want to, I'm just talking to you about interesting things related to what I'm lecturing on. The globin genes, you have embryonic globin genes that are expressed during the embryo, you have sort of infant globin genes that are expressed during childhood, and you have adult globin genes. So your hemoglobin is coded by different genes that are expressed developmentally at different times. So some of them are off and some of them are on at different stages of development. So it's a good model cluster of genes to study what's turning them on and off. And when they studied the globin genes, they then, all this hypersensitive terminology in the textbook comes from the globin gene work, because they identified these upstream enhancers that were hypersensitive when the genes were turned on. And that's how a lot of the textbook doesn't refer to the globin genes explicitly, but it's actually talking about the globin genes when it talks about hypersensitive sites and upstream enhancers. Okay? And that holds true for many other genes as well. So if you're studying gene expression in eukaryotes, you often have to include distant regulatory elements that you wouldn't have anticipated including. Okay? And then once you've got those distant regulatory elements in your test tube when you're doing your in vitro studies, you can then mimic the expression of the gene that you know occurs in cells. Okay? So again, that's really good evidence that these sites, that these bits of DNA you've included that are 50,000 base pairs away, you're now getting the correct response of that gene to the various factors. Okay? So there's lots of really good evidence that says these distant regulatory elements are involved, and the textbook goes with a looping argument to say this is how they're involved. Okay, but there are other arguments out there. Okay, so we're talking about turning genes on. So we're talking about positive transcription factors. So when we looked at bacteria, we were looking at, I've already said this, we're looking at one promoter per operon. So we had the one particular protein that was involved in the lac operon as an activator, and another protein that was involved as the repressor. And these were kind of unique and only worked at one promoter. In eukaryotes, we typically you'll notice that there are common transcription factors between different genes. So many different genes have the same transcription factor binding sites, but they've got very different expression profiles. So it's not the fact that it's got a binding site for a particular transcription factor, it's the combination of transcription factors. So there might be, say, 5 or 6 transcription factors which all bind within the vicinity of a promoter to turn it on. And there might be another promoter that has 3 or 4 of those binding sites in common, but then some additional binding sites. So it's this idea of a combination of transcription factors that regulate gene expression. Okay, so it's a bit more complex when you're doing these studies. So diverse promoters can often have similar sequences, but it's the specific combination of sequences that matter. All right, and some of these regulatory proteins are not just located at the promoter itself, but they can be the binding sites for these other factors can be more distant, and some transcription factors are found bound at many hundreds of promoters, whereas some transcription factors are only bound at a few promoters. So a lot of that work on combinatorial sort of binding of these different factors comes from fly as a model organism, and if ever you do fly genetics, it's really interesting to look at developmental expression of genes in fly, but we won't talk about that here. Okay, so when you look at these diagrams in the textbook, there are 3 broad classes of proteins involved in gene expression. You have the basal general transcription factors, which are the tartar box binding protein and the TF2Ds and all of those proteins. You've got the DNA binding transcription activators, so these bind directly to the DNA, and then you've got the co-activators, which it's more protein-protein interactions between different proteins. Okay, so the co-activators don't bind directly to the DNA. Okay, right, that's all right. So looking at gene expression, we have to recruit the tartar box binding protein and the basal transcription machinery at the promoter, and then we then need to activate gene expression through these positive transcription factors and co-activators. So there's a diagram here which we've seen before, but is a bit more, slightly more detail in this diagram the second time round. So this is the basal transcription machinery bound directly at the promoter region. Okay, so this is what has to be there for transcription to have a chance of operating. Once this is in place, with either close by or more distantly, you've got binding sites for transcription activators shown here. Okay, and then these transcription activators can be bound by co-activators, and then it's protein-protein interactions between these proteins and this big cloud of proteins we've discussed before, this mediator complex. So you've got protein-protein interactions here between these, and the or directly protein-protein interactions to the basal transcription machinery. So the transcription factors here don't interact directly. The transcription factors interact through these co-activators and this mediator complex. Okay, so in each so the other thing that's shown in this diagram here are these high mobility group proteins. I think someone should rename them. I think they're named high mobility group proteins because of the way they were purified. Because they're positively charged, they ran fast on mobility gels, so when people did experiments, these were high mobility proteins. It turns out they have a function of binding to DNA, and they're involved in this, possibly the looping of DNA and controlling some of that process. So a few overlays here. We have, for gene transcription in eukaryotes, we have the assembly of the pre-initiation complex and all of the factors required for the basal transcription machinery. We have the TF2D and a bunch of other things which can also need to bind. We've got transcription factors, activators bound to the DNA, and then we have co-activators bound to them. So we've got protein-protein interactions here and protein-DNA interactions for the activators. And then they can interact through the mediator complex. And also in eukaryotes we have high mobility group proteins as part of chromatin, and these proteins are thought to facilitate bending of the DNA and looping of the DNA. So just a few points on this mediator complex again. It's required for basal transcription levels, in other words, housekeeping genes that are on at a regular amount all the time, but it's also required for turning on regulated transcription, responsive transcription. The mediator binds to the carboxy terminal domain of the polymerase. So if you remember, this is our polymerase Pol II, and the Pol II has this carboxy terminal domain. We've talked about that before. It gets phosphorylated by proteins in the mediator, and when it becomes phosphorylated, it can then transcribe into the gene, and part of that process is mediated by these transcription activators. Oops. So phosphorylation of that carboxy terminal domain is important for the polymerase activity, and co-activator complexes also function near the tartar box. So, okay, so we've talked about remodeling of the nucleosome, meaning that the nucleosome can be shunted along away out of the promoter, or the nucleosome can be made to be more open so that the transcription machinery can get in there and start binding. You sort of have to try and think, well, what initiates the process? So one of the ways they're thinking is that some of these transcription activators, some of these proteins that bind, they have strong affinity to bind DNA. So they can bind to DNA more strongly than the histones can bind to the DNA. So some of these transcription activators can displace and start to open up the chromatin for other transcription factors to come in. So the binding of some of these transcription activators may then facilitate binding of others, leading to the gradual displacement of the nucleosome. Okay? So if you think about it, there's a certain affinity that the nucleosome has for the DNA, but there are some other factors that can bind more strongly to DNA and start to displace the nucleosome and allow other things to come into play. Some proteins can also act as repressor proteins. That's not surprising. So effectively, by modifying the structure of these proteins, you can enable them to act as activators or repressors. And this story is effectively talking about proteins such as steroid hormone receptors. So steroid hormones are cell signaling molecules that trigger transcription in cells. The steroid hormone can get directly into the cell, and the protein that is bound by these steroid hormones, it's a receptor, but it's also got a DNA binding domain. It's a transcription factor. So this thing that can bind to the hormone can be changed. It then causes a change in its structure, and then it becomes active as a DNA binding protein. So some of these proteins can act to repress transcription, but then a switch comes along, such as a steroid hormone, can bind to that transcription factor, which is also a receptor for the hormone, and it can change its conformation so it can act to activate transcription. So, this little diagram here. So this is one of these receptors. In the absence of a steroid hormone, it's acting as a repressor and sort of inhibiting some of these protein-protein interactions. The steroid hormone can come along, bind to the repressor, and change its structure, and then cause the repressor to bind to DNA or to change its protein-protein interactions. So just to summarize what I've been saying in those slides, the Pol-2 promoters have Tata boxes and initiator sequences which are at the promoter as well as distant regulatory elements not located at the promoter. Distant regulatory elements may come into play through DNA looping, and maybe these HMG proteins are involved in looping the DNA, and then this can bring these distant regulatory elements into play. And also we have the mediator complex, which can also bridge between these transcription activators that are not at the promoter and bring them into contact or proximity to the basal transcription machinery. So that's just summarizing basically what we're looking at in these diagrams here. And just to finish up the lecture, just a quick mentioning this idea of combinatorial control of gene expression. In other words, multiple transcription factors are having an effect on gene expression. So we've looked at the lac repressor as a transcription factor, and we showed the lac repressor binding to DNA through that helix turn helix structure, which got into the major groove to allow it to act as a transcription factor. People then started to think about other proteins that act as transcription factors. And when I was a yeast geneticist, when I was a kid, there was 120 transcription factors. Nowadays, people are saying there's 300 transcription factors. So things have changed in only 9 years. Okay? So we're discovering more and more functions of proteins all the time. And we now have about 300 transcription factors in yeast. In humans, we say current estimates are that we've got 3,000 transcription factors. So we've got 3,000 factors, which is an awful lot, except that we've got 30,000 genes. So obviously transcription factors have to bind to multiple genes, and it's the combination of factors that have an effect. The fact that transcription factors can work as dimers allows a greater range of transcription factors to be present. So even though we're saying there's only 3,000 transcription factors, there's actually more than 3,000 transcription factors. And the way we get more transcription factors from that set of genes is by different homodimers and heterodimers of transcription factors. So some transcription factors, such as AP1, can act as a homodimer or a heterodimer, and I'll explain that quickly in these slides. So AP1 is a well-known protein, eukaryotic transcription factor. It's actually made up of two proteins, but those proteins are both basic leucine zipper proteins. So this is the transcription factor. Along these alpha helices here are leucine residues, and they come into close proximity. They bind, and they sort of zip and unzip when they come together. So it's called a leucine zipper. And here's the DNA binding domains of this transcription factor. So if this is an important transcription factor, if it only worked as a, well, where's the diagram here? So here's the two components that make up the AP1 transcription factor, okay? And because there's two different components, you can get homodimers of either this or a heterodimer. So from these two independent halves of the transcription factor, you can make 3 different transcription factors. Okay? So if there are more than, say there's a family of these proteins, and we're showing 4 here, then effectively from these 4 gene products, we can get 10 transcription factors. Okay? So if we number these 1 to 4, so you've got 1, 2, 3, 4, you can have 1 and 1, 1 and 2, 1 and 3, 1 and 4, 2 and 2, 2 and 3, 2 and 4, 3 and 3, 3 and 4, and 4 and 4. You get 10. Okay? So you do the combinations, and 4 independent genes coming together to make a transcription factor can give you 10 different transcription factors. Okay? So the fact that you can get heterodimers of these components making up transcription factors gives you a much wider variety of transcription factors to allow for the fact that we've got 30,000 genes and a lot more genes. We thought we'd have more transcription factors than we actually do. Okay? And this is the same for signaling molecules in cells as well, but we won't go into that. All right. I'm not going to talk about fly gene regulation in fly, even though I'd love to, because it's a fantastic story and it's really interesting, but I won't. Okay, so this is the end of the lectures. So thank you for your attention, and good luck with the rest of the course. I guess Lisa will be back here next week.